Welcome from all corners of the continent and beyond. My name is Anafi Mataka, and I'll be your host today for our special COVID-19 session number 29. Amazing how far we have gone. This is a series of sessions that we have dedicated to diagnostics in the pandemic. Today, we are continuing with our current series on manufacturer dedicated sessions. And following the historic global launch of the rapid diagnostic test for antigen testing for COVID-19 by global partners, Global Fund, Unitaid, Africa CDC, FIND, and other partners, we continue to dedicate this session to one of the key tests that has been already listed by WHO EUL in the name Standard Q by SD Biosensor. We're going to look at the key details of what this test is really all about, how it works, the status of regulatory approvals, issues of performance, technical requirements, as well as some requirements for consumables, and also hopefully discuss what this means in supporting our testing programs for COVID-19, as well as fighting the pandemic. To do this, we have Mr. Cedric Joe, who is the International Project Coordinator for SD Biosensor, which is based in the Republic of South Korea. So we are grateful for him to be available at this time, which is probably very late at his end of the world. So Mr. Cedric, thank you very much for coming through, and I would like to hand over to you to share your screen and take us through the presentation. Colleagues, this presentation will be roughly 20 to 25 minutes, following which we are going to be taking questions and answers from the chat. So keep the chat box busy with questions, as we will address them as usual at the end of the session. Mr. Cedric, over to you. Thank you.
Thank you, Mr. Anafi, for a kind introduction. So, hello, everybody. I'm Cedric, and so I'm International Project Coordinator in SD Biosensors. I manage different collaboration projects within SD Biosensor, and I'm really thankful to ASLM for having this opportunity for us to present about our product for COVID-19 antigen RDT. And personally, it is my great pleasure to be invited by ASLM. Previously, in the same company, I was also regional sales manager charged of Western Africa, so mostly French-speaking countries. So it's very, very pleased. So I will stop my short introduction. I will continue with a company introduction. So SD Biosensor may not be a very familiar name, but we are actually based in South Korea. We are focused on developing and manufacturing different diagnostic solutions. So as you can see on the bottom of the screen, we produce different screening tests or at point of care level, like rapid diagnostic tests, but also glucometers on fluorescent immunoassay tests. But we also make different confirmatory tests, which are supposed to be used in laboratory settings, like ELISA tests and molecular point-of-care cases. So this is our brief history. So the company was founded in 2010, and that column is quite empty. I removed unnecessary information because from the foundation at the beginning, the company was more focusing on a glucometer business. So we actually started development of immunoassays around 2014. So as you see, we started developing the rapid test kits focusing on the issues for global health. And you can see we have obtained in 2014 the WHO EUAO. Also, we acquired that accordingly during the pandemic of Ebola virus. Following, we continued developing different diagnostic solutions for world health issues like MERS, coronavirus, Zika virus, etc. And recently, we have acquired some important certifications. So in this year, 2020, at the beginning of this year, we obtained a WHO pre-qualification for malaria rapid test, also HCV antibody rapid test. And we continued getting this approval for other type of assays like HIV and syphilis test on HIV RDTs. And we also, so for the COVID-19, very recently, we have obtained WHO EUAO approval for a standard Q COVID-19 antigen test. So the same tests are manufactured from South Korea. So our facility has all the necessary certifications like ISO, and also it has been audited by WHO PQ team previously for the pre-qualification process of malaria test and HIV test. And so the COVID-19 antigen test is also produced in the same facility. So now let's talk about more the COVID-19 antigen test itself. So when you open a kit, this is the contents that you will find out. So there are 25 test devices individually pouched in aluminum foil and 25 sterile swabs. So these swabs are used to collect the nasopharyngeal samples. And 25 extraction buffer solutions, and it's individually contained in extraction buffer tubes, which are sealed. And 25 nozzle cap, and this will be used further for application of the sample, but I will develop further how to use this nozzle cap. And obviously one instruction for use. And the sample type for now is compatible with a nasopharyngeal sample. However, we are working on making this kit compatible to nozzle swabs, so to improve the usability for operators, but also for patients, collecting nozzle swab will be less painful than collecting nasopharyngeal swab sample. For the testing volume, we'll explain the test procedure later, but it requires only 3 drops of a mixed sample with buffer solution. The testing time after applying the sample is between 15 and 30 minutes. So 15 minutes for the migration, but we recommend reading up to 30 minutes. And we do not recommend reading after 30 minutes, because it can show false result after 30 minutes. The storage and transportation temperature is room temperature, so it's stable up to 30 degrees Celsius. And operating temperature is the same, it's up to 30 degrees Celsius, but it has to be more than 15 Celsius. However, we are going to improve this stability, so we are soon to make this kit stable up to 40 Celsius with the further stability study that we are conducting. And the last line, you see our internal catalog number when you want to find the reference of this test. So this page shows the clinical evaluation results. And this is actually coming from an independent study organized by FIND. You can see the original report with the link, which is presented below the table. To explain further, it has been, so these two studies are showing here, so one from Brazil and Germany. In third column, you can see the overall average data. So let's go from the bottom. So in the bottom line, you can see the clinical specificity. We tested over out of 1,506 negative samples confirmed by PCR. The standard Q COVID-19 antigen test was accurate at 98.94%, so almost 99% of the specificity. For the sensitivity, you can see the second line from the bottom, and the overall sensitivity is 84.97%. So the total number of positive samples from two sites were 153 positive samples confirmed with PCR. And out of that number, our test was able to detect 130 samples as positive. However, in this total number, there are many different natures of specimens. So some specimens had very high CT values, so very low concentration, or some samples were from a patient having the symptom onset date very long ago. So you see the different sensitivities separated into different cutoffs. So we propose here showing 4 different kinds of sensitivity. So if you see, if you cut the cutoff, if you set the cutoff at CT value less than 33, the sensitivity in overall is 90.71%. If you set the cutoff of a CT value at 25 or less, it becomes 97.14%. And also, if you set the cutoff for the day, the number of days of the symptom onset, less than 7 days, it shows 87.88%. And for less than 3 days, it shows 90.24%. So all of this cutoff is from the report itself, and the rationale is from the WHO TPP that was released roughly a month ago. So it was suggesting the minimum acceptable criteria, like more than 80% at a concentration of a CT value less than 30 and less than 7 days, or 5 or 7 days of symptom onset days. So this test is acceptable according to the WHO criteria. Hence, this has been approved by WHO PICA team for the WHO EUL process. Now it shows the limit of detection. For the limit of detection, I just want to show that this test is actually compatible to use with the VTM or UTM samples with limited condition. However, when using transport media stored samples, there's inevitably sacrifice of the limit of detection. So we always recommend using fresh nasopharyngeal samples as the test kit is designed to be used at point of care setting. So when you want to start a test obviously the operators will need other accessories. So as you can see on the screen, the operators will need to wear the PPEs as described here, and disposable waste container for the hazardous material or contagious materials. And even though the test kit provides every necessary things to perform a test, however, a timer will be helpful to have to accurately see the time for the result. In this slide, you see on the left the conventional biosafety requirement environment. So this is something that will be required for testing of laboratory-based tests like PCR. So, however, this, the RTT is designed to be used at point of care. So in point of care setting, and this say biosafety level of facility may not be feasible. However, we tested internally that the virus is, the SARS-CoV-2 virus is inactive after 2 minutes once the sample is mixed with the buffer. So after 2 minutes, the virus, it becomes inactive in the buffer solution. So we think this is an important information for countries to think about the ways to adopt and set the guidelines for the biosafety requirements for the field operators. So once the testing is done, obviously we recommend putting everything in the waste box, and every contents of our kit are actually disposable. So absolutely, you have not, you must not use it, reuse any of the kits, any of the accessories of contents. So even if you use it wrongfully, please do not reuse it. Just discard it, because it is all of the cassettes are disposable cassettes. So these shows more of the contents that you are supposed to have in the kits. However, for the controls, the control solutions are currently not included in the test kit, but we have separate control kits, which contains tablets, lyophilized tablets of positive controls and negative controls. However, for better usability, we are working on including at least one of a positive and negative control swaps in the kits from the beginning. This will be applied pretty soon. So, as I mentioned at the beginning, the storage condition is between 2 and 30 Celsius room temperature, but we are in process for improving this stability. So we hope reaching this kit to be stable up to 40 Celsius very soon, probably by end year or beginning of next year. Also, when you collect the sample from a nasopharyngeal swab, and we recommend using the sample immediately on site. So we do not recommend storing the specimen too long a time. This is the precautions figuring in our instruction for use. So this is quite a basic information to not to do, or yeah. So I'll probably skip this slide faster. And in our kit, obviously the package will show the manufacturing date, expiry date, and lot number. But since the test cassettes are pouched individually, the individual aluminum pouch will show the same information, such as lot number, manufacturing dates, and expiry dates. And the extraction buffer tubes are also sealed in another aluminum pouch, and on that pouch you will also see the manufacturing date and expiry date. So please absolutely check and recheck the expiry date before using the kit. And when you open this aluminum pouch, you can take out the test device. And in the same pouch, you will find a desiccant that is included together. So you have to check the color of the desiccant. If the desiccant color is yellow, then it's fine to go. But if the color is green, it means that it has been affected by humidity. So please do not use that kit, and please open another test strip. So to perform the test, you collect the nasopharyngeal sample with the provided swabs in the kit, like it shows in the slides. And you put it immediately in the buffer tubes. So buffer tubes are individually filled, and it is sealed with foil cover. So remove the foil cover from the tube and put the swab in the tube. And then you start mixing it with the buffer solution. So stir the swab and squeeze the buffer tube. So the buffer tube is made with quite flexible material, so it is easy to squeeze with fingers. Once mixing is done, assemble the tube with the nozzle cap, which is also provided in the test kit. This page shows how to test with VTM or UTM stored sample. However, I mentioned that the test can be compatible to VTM, but in limited condition. And actually, there are various manufacturers making transport media, and we have only validated a few number of that. So in the table on the left side, you see 3 different marks of the transport media, and these are the transport media that we recommend using. So any of other brands or manufacturers' product may not be compatible to be used with ours. This shows some precaution when handling the sample. So the item 2 and 3 will be concerning the transport media, but item number 1 is showing that the sample, the mixed sample, is stable up to an hour in room temperature and up to 4 hours in low temperature settings. So this is why we recommend using the fresh nasopharyngeal sample to be used immediately inside. It also shows basic recommendations. So the test strip will be require 3 drops of mixed sample. It can accept 4, even 5 drops is okay, but putting less than 3 drops might be problematic because the migration might not be complete. So we recommend putting sufficient volume, so at least 3 drops or 3 or 4 drops. But in our official instruction, we recommend putting 3 drops of a mixed sample, which will be around 90 microliter. So as this shows, apply 3 drops of mixed samples and wait for 15 minutes. So 15 minutes and 30 minutes. So you better not read the test result after 30 minutes because it may show false result, especially false positive result. So once the migration is done, you can interpret the test result as indicated in the slide. So it can be interpreted either positive, negative, or invalid. So next to the line, you see a small letter C or T. So C for control line and T for test line. And when the test line is on, it means the test result is positive. So these are some reasons that may have a false positive or invalid result. So like saturation of specimen or the sample volume. Sometimes it may cross-react with other substances. This so there are 2 ways of control for this rapid diagnostic test. So first control is the control line, which shows that the migration is done correctly, and this is already included on the strip. So it is this is the first thing the operators have to check to see if the completed test shows line C or not. If it does not show any line on C, that means the test result is invalid anyways. However, we also have a control solution, as I mentioned. So at the moment, this is sold as separate pack containing 10 positive and 10 negative tablets. So this the operators can use this tablet with the test kits. So the tablet is to be mixed with a buffer solution and to be applied on the test strips. However, as I mentioned, these controls will be soon included in the test kit itself. We are also in development of mobile application to manage the data. So at the moment, as it is not launched, I cannot show how it works, but soon we will have opportunity to present it for different actors in the healthcare field. For now, this standard Q COVID-19 antigen test is officially certified by WHO, AUL, and CE mark and TGA from Australia. And you see some names of countries where the kit is already registered among 8 others. So our goal for entire African continent is registering this kit to every country or where the registration is necessary to enter to the country, so that there will be not missing country from any. So we will not miss any country to be supplied. And in most of countries, we have a representative there. So if there's any need for training or after-sales service, our distributors can help to assist the local end users. The troubleshooting. Basic steps, and this information is from the IFU, in my understanding. So yeah, so obviously it should verify the labeling. That means they verify the expiry date, lot number, etc. That are matching. And if the procedure has been followed correctly, and if the test result is suspicious, you can rerun the test with other device, and you can repeat with another operator, and you can also go to collect the same specimen and go to the confirmatory test for validation. And now, where to find our test? So after our commitment that we made to the global public health community, we created a dedicated page on our website. So you see the domain and the web address here. And every public entities are eligible to make direct order to SD Biosensor headquarters. So please use this page to make an inquiry to us, and we'll be responsive. And you can also contact the distributor located in your region or in your country. For now, we do not publicly open the contacts of our distributor per country, but if you make any inquiry through the page that I presented here, we can also connect you to our distributor for better assistance. And there's other ways, like this kit is already on catalog of different markets, like WHO catalog, Global Fund, UNICEF, and Africa Medical Supply Platform, etc. So you can go to these platforms to make an order of standard acute COVID-19 test. So thank you. This is end of my presentation. If there's any question, I'd like to answer. Yeah, thank you.
Okay, thank you very much. Sorry, I think I had a glitch somehow. Cedric, for the wonderful presentation, quite informative indeed. So it's now time where we take questions from the chat, and wow, lots and lots of questions indeed. As we try to take the questions. All types of questions. I think maybe let me start with the issues of sample types. I think quite a lot of questions on sample types. Cedric, just to make sure that we clarify what is the recommended sample type. I know you might have touched it, but for the benefit of those that joined later on, what is the recommended sample type for this current version of the test? And are there any plans or any studies going on to see if other alternate test sample types can be used?
Yes, Nafi. So at this moment, this test is claimed to use nasopharyngeal samples. So this is why we provide the swabs that are dedicated to collect the fresh nasopharyngeal sample from patient nose. And I mentioned briefly, this may be compatible to certain models of transport media. However, given the limited number of this recommended transport media, you may not be able to find these models necessary. And the third one is that we are under process of validation of a new sample type, which will be using nozzle swab. So the operators can collect the sample from lower nasal cavity from patients, which will be much easier and less painful for patients. So we believe this validation will be finished within a month, and we'll be presenting this data to regulatory authorities to get final approval. So we hope applying this new sample type before end of the year.
Thank you once again, Cedric. Moving on, I think quite a number of tests, quite a number of questions around issues of performance, and I think many people were touching around sensitivities. Just to pick a few questions, some of them are actually clarifying, like for example, Dennis says, how is the sensitivity different from clinical sensitivity? After having seen quite a number of those. So I think it's also important for us to define analytical sensitivity as well as clinical sensitivity as well. The McPoy Okoye says, how is clinical sensitivity defined as in days from onset? So maybe let's start from there, then we take on a little bit more on sensitivity.
Yes, maybe I might go back to the slides of the table. So as I presented, this data is generated by independent study organized by FIND. So the sites of study, Brazil was the University of Rio de Janeiro, and Germany from University of Heidelberg. And all of the samples were collected prospectively. So these samples were freshly collected with our nasopharyngeal swabs that are included in the kit. And then the same patient specimen were tested against the PCR, and WHO or EU cleared the PCRs in Germany and Brazil. So this is all controlled and validated data confirmed with PCR. And in terms of sensitivity, when we present a general clinical sensitivity, so let's see the second line from the bottom. The Germany data shows a 76.6, and the overall average data is showing 84.97%. But if you look at the data with the more meaningful cutoff, so with the CT values, so the rationale for for the CT values is when the CT value is too high, the viral load is probably quite low, so the patient may not be immediately contagious. So the priority is getting the patients already contagious and ill quite seriously. So obviously, the platform itself will have its limitation because this is point of care rapid tests are not laboratory based PCR tests. So obviously the performance will not be matched to the PCR performance, but this is somewhat very useful for point of care setting, especially in remote area when where we can't collect the sample and go to the laboratory, which is too far away, for example. So to make a clinical decision quickly, this is quite a good test to assist the clinical decision on site at point of care. This is the purpose of this test existence, and hence the WHO TPP suggested using it in that setting as well.
Yeah, thank you, Cedric. Yes, indeed. I think it suggests values of about 80 and then specificities above, I think, 90, around 97. So also to just for the benefit of some of the questions that came through in trying to clarify whether analytical sensitivities, like asking if that assay is targeting a certain element or target, can the assay detect it? Whereas the clinical sensitivity is sort of referring to the patient to say if the patient is sick with that disease, can the test diagnose it? I think maybe that also may be very useful for us to clarify to our audience. Then there were actually questions around, I think, the performance of evaluations and what kind of sample sizes were used. Maybe just to quickly clarify that. Yes, go ahead.
Yeah, sorry. So the question was the sample size of this clinical evaluation?
Yeah.
Okay, yeah, so it's on the table. Let me explain. So for Brazil side, the specificity was tested over 294 negative samples that were verified with PCR. And from Germany, the total specificity size was 1,212 negative samples that are verified with PCR. And for the positive samples from Brazil, the total positive samples that we found in the study was 106 positive cases verified with PCR. And from Germany, 47 positive cases, actually positive cases that we found with PCR confirmation. So the total number of positive sample was 153, and on that number, our test found 130 positive results. So the sensitivity is calculated upon it.
Thanks again, Cedric. Yeah, just picking up the question from Fatim Jallo, I think still around the issues of performance and how this test can be used. She says, can you clarify on the need to confirm positive test results as per the instruction for use? And I just want to link that to a comment from Kin O, who is also saying they have used the SD biosensor widely in his or her country a few years ago and a few weeks ago, sorry. And it has been helping to prevent the backlog of samples sent for PCR. According to her, that this is very useful, but a few false negative samples have been found beyond a week-old samples. And these have been suggested to be confirmed by PCR. So maybe just to clarify on the need to confirm a positive result. Is there a need to confirm a positive result?
So to reconfirm the test results from the field, but I think this is entirely on the decision of the settings of each of countries. So the prevalence is different on the situation is different country by country. So so whether or not go to the confirmation after a positive or after negative, but this is something that's beyond our recommendation. So this test aims to give screening test results, and it is up to the decision of the healthcare authorities or the authorities who's charged of the decisional responsibility there to decide whether or not the further confirmation is needed.
Thank you once again for that. I can acknowledge, I think some questions asking for slides. Indeed, this will be shared as usual. So moving on, I think there were quite a number of questions around safety, and I'll pick a representative one from Master Chisale. Master says, from the presentation, it has been mentioned that COVID-19 is inactivated after 2 minutes when the buffer has been added. What are the safety measures to be considered since this is a point of care test? Any study results that show possibility of infection of lab staff? And I think there were quite a number of questions asking around that 2-minute interval for inactivation.
Yes, so this is coming from an internal study. So the virus is inactive after 2 minutes from the mixture of buffer solutions. So the 2-minute time is fairly the time after applying, and so this is quite short in the field when we count 2 minutes. So we think this makes a lot of options to healthcare authority to have a more or less flexible biosafety guideline based on these factors. So obviously we are not here to give an absolute guideline for the biosafety, because the biosafety is to be set and decided by the local healthcare authorities. And here we just give an information that will be useful for the healthcare authority decision makers to consider this information to further develop the safety guideline. So, so today we simply give the information on. We do not want to make confusion among the operators for having certain guideline or not, because the situation might be different from country to another.
Thank you once again. So just to also make sure that this is also clear, Damitu Negashi is asking whether this kit is registered on the WHO or Global Fund PQ list. I think it's the WHO EUL list. In terms of Global Fund, I saw Fatim was here. I don't know whether she can be able to also respond to whether this has been already listed. Fatim, are you able to just quickly respond to that?
Yes, Anafi, it is listed.
Thank you, Fatim, for clarifying that as well. So indeed, it is listed. And there was also a question on pricing. We know within the global launch, I think these were estimated to be at $5 a test. Hopefully that will also continue to go down as well. Moving on, I think we'll go back again to the questions around samples, and Ahmed is asking whether the kit shows the same result using VTM or directly into extraction buffer. Yes, Joe.
Yes, so when you see this slide, yeah, so I skipped actually how to use the VTM samples with our kits precisely. But with the pipette, you collect 350 microliter of the viral liquid from the transport media. And you mix it with the buffer solution, which is provided in our kit directly. So the ratio is 1:1 almost. So you mix it properly on, you apply after assembling the noise cap, the same number of drops. So 3 drops from the mixture of viral liquid on the and our buffer solution. Yeah. However, the concentration of a virus will be significantly lower than mixing with the fresh swab in general with our experience. Hence, the LOD was quite less good than the fresh samples.
Yes. All right. Thank you very much. I think there is also a question which is from Angela Amayo, who is asking, should the test be for asymptomatic or symptomatic patients? I find this interesting also because she continues to talk about the recommended clinical utility is important and perhaps an algorithm for the use of the assay. And also to link that, this speaks to the question from Mason Lucas, who is asking how long it will take for this test kit to detect positivity.
Yes, so the infection stage depends on the patient's health status, the conditions. So we can tell that it is accurate for everybody. So number of days is only indicated. So, but we suspect from the potential infection date, from the penetration of virus into the human body, the antigen is supposed to be detectable from day 2 of a potential infection date. However, from the symptom onset date, when the symptom is already visible, it is supposed to be detectable with our antigen test too. And as regards the asymptomatic patients, we've been seeing that this kit was able to detect asymptomatic patients as well. So the key is whether or not the patient has enough virus antigen in the nasopharyngeal or not. So regardless, because with the COVID-19, there are so many patients not showing the symptoms visibly. So many of the asymptomatic patients are actually detectable with the kits. But in our study, this was not designed with asymptomatic patients specifically. So we know this is technically possible, and probably we'll be presenting other data sets to show this. So this is asymptomatic patient is technically detectable, and yes, from the early stage of infection, yeah, it is supposed to detect the patient at early stage too.
All right, thank you very much. I also want to pick this question, I think along the same lines, from Max Pina. And I picked this because we also discussed about it about 2 days ago. The CT values tested, I think for the corresponding samples here, must be obvious positive cases. And she wishes to know, or he wishes to know, the performance of this RDT when CT values between 35 and 38.
Yeah, so as I mentioned, the higher CT values have quite low concentration of virus. So it is indeed very challenging for the lateral flow assay format to detect this range very accurately. So this is something in this data set, it doesn't show this data, but because very few samples were in that range of CT value. So today, yeah, unfortunately, I don't have in my hand other data, so I can't accurately tell numerically what was the sensitivity or specificity in that range. Sorry.
All right. Thanks a lot again, Cedric. An interesting also question came through. I think you clarified that during the presentation about whether controls were included. And I think you said they are separate. And then Felix Nyakwawa is asking, any consideration to maintain the price if controls will be included in the test kit in the near future?
Yes. So as I said, the control kit for now is not included in the test kit, but we're going to soon apply test kits to get the control swaps inside. So one positive control and one negative control. And but we will definitely not revise the price upon this, because this is something we have a commitment to the global community. So this this change will not have any impact on the price increases. So we will keep at least the same price from even after the change.
Thank you again. There is a question here which is very pertinent, and it's from Ashini Fernando. And she is asking, do you have any recommendations for in-country verification of these tests? You may answer this, and I can also call upon any people in the chat to also respond to this, because this is one of the key issues that we have always tried to address.
Sorry, Nafi, can I see the question?
Ashini, do you have recommendations for in-country verifications? Whilst you are checking it, I can also attempt to do that. I think verification is just simple verifications as part of good laboratory practice is okay. Though, of course, I think people have said, I think as long as these tests, I think, have been evaluated several times, and we continue to gather this information, I think it may be important for people to have ongoing verifications as they are going through, so much so that we do not, I think, stifle the introduction of technology that has had some form of evaluation at central levels.
Yeah. Well, I see that, but unfortunately, I can't answer to that question today. Yeah.
Thank you. Yes, so I think the issue of controls is really raising interesting concerns and discussions. Then, such as Augustine Onyegala, who is asking whether this is scientifically valid to say a kit without controls for a sensitive test such as COVID-19. Any comment?
Yes, this is why we are hurrying to applying the inclusion of controls in the kit. But as I mentioned, the control kits are in a separate package. So temporarily, we're thinking probably presenting control kits in a separate pack format, but very temporary. But soon the operators will see the kits delivered all in one package, so with controls. But we know that having the controls in the test kit is the best option. So we are going to make this change very quickly. But in between, we can also offer the control packs in a separate package for temporary option.
Thank you once again, Cedric. Yeah, we have reached the top of the hour, and I just want to ask you to just give a few seconds in terms of just your parting words when in terms of the test and what is capable on what we discussed today. Yes, Cedric, any closing remarks?
Sorry, so thank you very much. I hope I gave enough clarification on the test kit, how to use, how to include international strategy for the response to COVID-19. So we'll be very responsible to the market's demand. So any suggestions, like I heard today, I noticed that a quick inclusion of a control is a high priority. So we'll be focusing more effort on that. So thank you very much again for this opportunity. And I'll be joinable by email. So any questions, you can address me directly to my email address. Later on, I will share definitely this PPT to ASLM so that ASLM can share this contents to everybody who needs. Thank you very much.
Thank you, Cedric. Indeed, we'll be glad to share the PPT and any other related materials that you share. Colleagues, thank you once again for attending this session. There are loads of questions. Indeed, there are some that we haven't. So we will reach out to Cedric to help us respond to some of those questions indeed. Just want to remind you that lots of questions that are coming through, and they continue to refine the package of RDT antigen for SARS-CoV-2 that has been developed by ASLM and partners. So this will be launched very soon, and keep watching, and hopefully we should be getting out to you soon. I also want to be sharing with you just now, for those that have been asking about certificates, on the screen coming shortly will be the code for you to be able to share, to go to the ASLM Academy and be able to download the certificate, and also be able to look at some of the sessions that we have held in the past, of which you will also be able to download the certificate as well. So we will continue with these special sessions coming through. There were questions around the issues of the price of testing, how this is going to be procured. And we're also hoping that we can also have a session talking about procurement mechanisms as well as funding. We have also not forgotten molecular testing, and we are also looking forward to be bringing a session where we talk about pooling of SARS-CoV-2 molecular testing as well. Ladies and gentlemen, it's been a wonderful session, and I wish you a wonderful weekend as we go. For me, this is goodbye. Thank you.